It's just a science project.
Silent breed is people!
You know, a doctor friend once said the same thing to me. Frankenstein was his name.
It's alive! It's alive! It's alive!
That sounds like something out of science fiction.
Please explain to me the scientific nature of the whammy.
We live in a spaceship, dear.
So? Yes, science!
Program complete. Enter when ready.
Hello and welcome to episode 211 of Science on Top. I'm Ed Brown and I'm joined by Penny Dumsday.
Hello.
Lucas Randall.
Hello.
Dr. Mick Vag.
Hey, guys.
And Bad Santa, Dr. Shane Joseph.
Hello. I was wondering what the hell you were planning.
I always have to throw in something about you not liking Christmas, because it's Christmas now. And this is our last show of 2015, and that means it's time to look back and remember some of the stories that we found particularly interesting. But before we do that, I want to mention some, breaking news. In the last 2 weeks, more than 80 children at a primary school here in Melbourne have contracted chickenpox. Now, Mick, that's a quarter of the school's population afflicted with a vaccine-preventable disease.
That's right, Ed, and that's enough to make it a public health hazard, really. And I think part of that is the false tolerance, I suppose, that the school is showing to vaccine deniers or vaccine refusers or the vaccine wary. But, this is what happens. Chickenpox virus, the varicella virus, just doesn't care. It'll just do what it does. And this is what's happened here. In fact, my daughter had a sleepover at a birthday party, and just across the road, and they live probably about 400 metres, 500 metres from this primary school. And there's a crystal healer in the house opposite them who has been getting a lot of visits lately, apparently, from that school.
A crystal healer?
A crystal healer.
She heals with crystals, or she makes crystals feel better?
She heals by the agency of crystals.
Wow.
Of course she does. Yeah.
So when you say she heals by the agency of crystals, do you mean that she's full of crap?
No, she's just the conduit, and the crystals help her focus her healing energy. It's the universe.
Try and follow.
So she's full of crap.
Yeah, duh. Yeah. But on a serious note, though, I just sort of did a little bit of jotting down of exactly what this sort of an outbreak means for the kids that are involved, and just to show why so many of us sort of sciencey types get so wound up about this vaccine denial stuff, is that if we round that 80 kids up to, say, 100, of that group of 100 kids, during their illness, about 60 of those 100 kids are going to get evidence of cerebellar inflammation, and that is they'll become, discoordinated or have trouble coordinating their movements properly. About 15 of those kids will show some evidence of encephalitis, which is generalised inflammation of the brain tissue. 2 of those 100 kids will show signs of meningitis, and 1 will develop a facial palsy, that is sort of weakness or absence of movement in facial muscles. Most of those will recover, but about 2 or 3 of them will have permanent neurological damage from that episode. So of those 100 kids, 38 will get infectious complications. 26 of those 38 will get skin infections, and of those 26, 8 of them will be a type of necrotizing fasciitis, or the old flesh-eating bacteria type infection. 4 of them will get septic arthritis, that is a bacterial infection of joints which can permanently damage the joint, and 1 of them will get pneumonia. Of those 100 kids, 2 of them will actually get post-herpetic neuralgia, which is a type of nerve pain that you get following nerve damage. And 20 to 30 of those 100 kids in later life will develop post-herpetic neuralgia, and half of those that get post-herpetic neuralgia will get long-term neuropathic pain, which is very difficult to treat. So this is not a trivial incident for these kids, and it's highly likely to have long-term serious consequences for a fair proportion of them.
Because I think a lot of us might remember getting chickenpox when we were kids, and I remember I just had red sores break out, and I was really itchy for a few days. I obviously got a mild case of it, but there's big risk of further complications, as you say. And also, isn't this what causes shingles later on in life?
Yeah, and that's exactly right. Yeah, that's post-herpetic neuralgia, and so shingles is the reactivation infection that you get down the track. And this is the other really important thing that people don't understand, is that wild-type chickenpox virus or wild-type varicella is about 4 times more likely to give you the post-herpetic neuralgia nerve damage pain afterwards compared to the vaccine version of it, because the vaccine, of course, is an attenuated virus. So if you get your kids vaccinated against chickenpox with the attenuated virus, first of all, you're likely to protect them from a severe primary infection, but you're also potentially preventing them from having, decades of severe nerve pain, because they're much less likely to get that nerve damage pain afterwards.
I'm just sick of more and more stories coming out with this sort of thing happening, and we say, vaccinate your kids, and it seems to not happen. People are still finding reasons to object to it. They're still reading nonsense on the internet and using that as scientific knowledge.
They're doubling down on it in their positions,
There's such they've invested so much in it, in their objections, that they're not really swayed by all of the things that Mick just said. Unfortunately, they've got these beliefs now that are quite firmly entrenched, and this is something that I've had some exposure to myself through my wife's kinder. There's quite a certainly a not insignificant percentage of the kinder population in an area that's fairly well off. Most of them are fairly well educated, and that seems to be the exact type of member of society who's really on board with this conscientious objection. And yeah, it really doesn't matter what you say or what facts are laid out in front of people who have these objections. They just won't budge. And yeah, I don't think anyone really has an answer for this yet.
I remember Joe Alabaster posted up something quite interesting on her Facebook about this. It's about sort of empathy for anti-vaxxers in terms of, not empathy, but maybe that's the wrong word, but in terms of trying to browbeat a lot of these people into, with facts and figures and sort of to call them, resorting to sort of strong language is probably one of the best ways to make them even more entrenched in their views.
Absolutely.
And
that's defensive.
And it's a real problem, because, we know we're right in this case, and the science is there. It's there's no question. But for these people, it's a life calling.
It's the fight fire with fire technique, where you throw all your arguments up to refute all their arguments, and it leaves both of you going, well, you're wrong, I'm right, and doubling down. You need to sort of pick apart the little argument here, a little argument there, and hope that enough of that smaller argument, I guess, builds up to at least make someone rethink their views and to question themselves.
Yeah, there's a technique we use a lot at work called motivational interviewing. And one of the tenets of motivational interviewing is that people are much more likely to change if they talk themselves into it. Mm-hmm. So what we have to have a conversation with these people in language that forces them to talk themselves into the position that we want them to have. And there are a number of techniques you can use for doing this, but one of my favourite ones is what you call developing discrepancy. So you don't necessarily point out all the flaws. You say something that will lead them to say, oh, hang on, that's not what I've heard, and then you just go, oh, okay. and then they will want to ask you about that, because they think, well, maybe if I'm wrong, and you just don't browbeat them about it. You just go, oh, okay, yeah, isn't that interesting? and bit by bit, you can bring them along. It takes time, but it's easily the most successful way of changing people's strongly held opinions. Because you're exactly right. The more you force them to defend themselves personally in that conversation, they may talk themselves into a position that is stronger than the one they actually hold, and then we've not only not convinced them, but we've actually made it worse.
Yeah, and you can have this. You can say exactly the same thing when it comes to, say, climate change.
Oh yeah.
People are. You can't reason them out of this position because they didn't reason themselves into it. They're not there for they haven't applied a logic to get there, and it is all emotive, and that's why as you say, you have to let them sort of make their own path. And then the obvious next part of the conversation is, well, is it worth having the conversation for the benefit of the people who are on the fence or who have only heard a little bit and they're just not quite sure what the truth is?
Well, and the other problem with this Brunswick West example is that I'm going to take a bet that a lot of these people whose kids are affected by this are going to say, "Oh, it's just chickenpox. It's okay.
Yeah,
it's it's part of their natural immunity. They'll build it up. But we don't need no stinking vaccines because our child will be immune." But as Mick said, when it's this prevalent and it can spread through a population this quickly,
And sooner or later, someone who's not well equipped immunologically to deal with it is going to get it, and it is a potentially fatal disease.
Those figures I gave were for immunocompetent children.
Really?
Wow.
So the figures for immunocompromised people and children are much worse, obviously. And when you've had to be involved with treating a disseminated varicella zoster infection in a pregnant woman in ICU, you don't forget that in a hurry. And it's horrific to see the damage this virus can do if it gets off the leash. So, in order to protect the vulnerable people in the community, people should just do the right thing.
Is part of, I think this has been brought up before, but is part of the fact that these sorts of this vaccine sort of total disregard for it, a lot of it might have to do with the fact that we just don't get these diseases much anymore. Sure. Like because of the vaccines have worked and therefore people have become complacent, and like all of a sudden chickenpox is just considered a fairly benign infection.
Yeah. It's so true. I mean, I've worked a lot with polio survivors, obviously in pain and rehabilitation. There's still a few of them around, still quite a few of them around, and there's not one of them that is not been traumatised by whatever the disease has done to them. That's one thing, but the social stigma and the isolation and the constant feeling of having to prove that you're at least as good as everyone else, if not better. And, there's a saying in rehabilitation that if you want something done properly, tell a polio survivor they can't do it, because then they will do it perfectly. Because these diseases have gone away largely in Australia. In fact, polio has almost gone from the world, and that would be a magnificent day because it has produced a huge amount of suffering that goes on for decades after you get the disease. And I think it's true that we've forgotten a lot of this stuff.
So, Mick, is there a danger when when these diseases, as you say, start to get hold? Is there a danger of mutation and then an undermining of the vaccines that we have?
It seems that I think that's very much a virus by virus thing, and Shane might know something about this compared to me, but Mel Thompson would probably know as well. But I think it largely depends on the virus and its capacity for mutation. As far as I understand it, polio doesn't mutate a lot, and neither does varicella, and that's one of the reasons those are very vaccine-susceptible diseases.
Right.
Well, it's a bit like the flu, which is a very, 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 very good at changing itself every season. So it's, that's the difference. Like Nick said, something like polio is very stable, so it's not going to change much, and therefore it's a much better candidate for total eradication. Whereas the flu is just going to keep changing every season. So, but the important thing is to keep the vaccines up to date.
And the other reason I wanted to mention the vaccine, I'm going to use the word debate, even though it's not really a debate, was to tie it back in with the looking back at the year. We started the year off with more than 130 cases of measles linked to visitors to Disneyland. And there's been a lot of outbreaks throughout the year, but it's just, like I said, I'm getting sick of talking about it all the time because it's just so important that we need to get vaccination happening. But anyway, shall we look at some of the astronomy stories, the space-related stuff? Lucas, we've gone to Ceres. We've sent, is it the Dawn probe to Ceres?
That's it, yep.
And we found these bright white spots. There's a number of them. And we're fairly certain that they're SoT.
Yeah, which was when we covered this earlier in the year, and I think it's come up a couple of times since in sort of, we've referred to it with other stories. But only in the last week, there were some news stories about this where some NASA scientists have said they're now very, very, very sure that these are basically salts that are left over from a salt basically a slushy brine sort of mix, similar, I guess, to what we've talked about before with some of Jupiter's moons. It does appear that Ceres has got this ice containing salts under its crust, and then if something disturbs the crust, like some sort of impact, then that would be exposed, and over time the ice would basically boil off and disappear, but it would leave behind the salts. And as we know on Earth, when in these salt lakes, they can be incredibly white. They can also, if they've got bacteria in them, they can be other colours. But obviously, if we found bacteria on them, that would be real news. But in this case, yeah, that's definitely seems to be what it is. And this has basically come out of the, because the Dawn craft right now is in its final stages of this spiralling down orbit as it gets closer and closer to Ceres. So it's able to do more and more analysis and as it gets closer to these things. And we start to see some awesome footage as well, some of the videos that are available.
Yes.
Time-lapse images put together. There's 3D renderings of Ceres as well, so you can see the whole sort of spheroid, if you like. It's very, very cool. But yeah, they're pretty now sure that it's that, and it's not. I remember when we first saw these things, it was definitely a collective case of what the, because they were so bright, and of course everyone's like, white things, aliens.
Yeah, landing lights.
That's it. Yeah. But yeah, that appears not to be the case. But yeah, it's always, it's amazing though when you look at the stories that we've covered this year in astronomy that have caused the biggest interest and excitement. They're all the probes. It's all the probe stuff that we're doing. But that's mainly, of course, because we're not sending humans anywhere other than the space station.
But also, I think it's because there's mysteries that we're solving, and we also there was the mystery of why there are less volatile molecules on the moon than there are here on Earth.
Yes.
And that was to do with the formation of the moon, wasn't it?
Yeah, that was one that we covered not too long ago. Related, once again, there was no new data in any sense. It was just a new way of looking at what we already knew, and some new models which applied to that information showed that it's entirely feasible for the moon to form in such a way that, part of it part of it formed in a volatile rich environment, and then as it gradually moved away from the Earth, the other part of it formed in a volatile, low volatile environment, if you like. And that sort of explains why there's this strange characteristic of the moon, which has been used in the past as one of the potential arguments against the impactor theory of the formation of the moon. But now, when we construct these models to explain what we see, when we can no longer poke holes in what we see, that becomes the accepted theory. And so that's even more so the case now with that observation.
Yeah, it's the ongoing amassing of theories and evidence to support your hypothesis and how we look at things.
I think you mentioned that it's that we've solved a few problems this year, but what's even more exciting is we've discovered a whole lot of new interesting problems in astronomy, and that's what science is all about.
Absolutely, and this will always be the case as we send more and more instruments to more and more places, and we apply, like in the case of telescopes, for example, new wavelengths to observations. We've got James West coming, which will be James Webb.
What are you on about?
James West.
James Webb.
James West. What the hell are you doing?
James Webb. It's because it's the James Webb Space Telescope. So I kind of was picturing the.
I'm just trying to wonder how James West fumed, but.
So with James Webb coming up, it's going to open up a whole new wavelength to us for space-based observations, which could be very, very interesting indeed. But as you say, Mick, the more we send out there, the more we learn, and the more we learn about ourselves as well, because there are certain things that we've long discussed, like, for example, terraforming Mars. This has been a long-held possibility for mankind's future, that we could go and terraform Mars so that it becomes more appropriate for mankind to live on it. But what we're starting to find out through not just astronomy, but a whole lot of Earth-based science as well, is that the Earth has a very, very fragile ecosphere. Its atmosphere is very easily pushed in other directions, and we can see from geology and biology and a whole lot of other things that we've used on Earth that keeping Earth in just this right sort of Goldilocks situation, if you like, is possibly going to be a much bigger challenge for us than we thought. And then you've got to start to think to yourself, well, how hard would it actually be to turn another planet into an Earth-like atmosphere when we can't even keep our planet in the right frame? So, we really need these discoveries to drive us forward, because sooner or later we're gonna need to take some action about our own planet.
Well, one planet that we've certainly learned a lot about this year, and I think for me it was the highlight of all the astronomy and space stories that we covered, was the flyby of Pluto by New Horizons, which talk about finding new things and new mysteries. It was a goldmine for that. Everything from ice volcanoes to a thin atmosphere, and the leaching of dust particles from Pluto to its moon Charon. It's been a huge story just from that point of view alone.
If scientists could have written a script for the most surprises they could get out of one mission, they would have failed to come up with this story, because it's just too extreme. It's just incredible. We knew so very little about Pluto previously. Up until this mission, it has always just been a very blurred circle. That's all we've really known about it.
I haven't seen it, but I'm assuming that there's a BuzzFeed article. Everything you thought you knew about Pluto is wrong.
No, won't it be 12 things you thought you knew about? But we now have got very detailed maps of Pluto's surface, some incredible imagery, and we've only got just a fraction back of the data that's going to come back from Dawn. We've also, not Dawn, we didn't divert Dawn from New Horizons. And New Horizons, of course, isn't finished. It's still sending its data back to us, but it's also heading off to investigate another object off in the Kuiper Belt. So,
if it gets the funding.
If it gets the funding, but obviously the funding now is more about the labour effort and just maintaining an instrument lock on. But the amazing thing with this probe is it just delivered so much more than they expected it to. I know there was some discussion earlier on in the planning stage of this mission that how much are we really going to learn with a flyby? Come on. When you think back to some of the other famous flyby missions out there, the Voyager missions, for example, spring to mind. And these are probes that are still going, still talking to us from the 70s. It's just incredible. But this was just an incredible mission, and it was a good one to live through with my kids so that they could see knowledge being born for us, which is very exciting, rather than just learning about stuff that their grandfather's generation did all the time.
I looked for ages and ages and ages when I was a kid at the National Geographic spreads on Voyager's flybys of Saturn and Jupiter and all the moons, and that was such a massive part of my childhood. And now for my kids being able to see things like the 7 minutes of terror thing in real time, and the the flyby with New Horizons, it's exciting and it's extraordinary times.
Yeah, the 7 minutes of terror. We were there, weren't we, Ed? Not in Mars.
Well, we were in Canberra at the Deep Space Communications Centre.
Yeah, and we were there for that period because they were involved in relay for the telemetry and stuff for that, and it was just amazing. The atmosphere was fantastic.
There was also a lot of medical stories. I think, Mick, you were particularly impressed with the stem cell treatment that has halted the progression of MS in 91% of patients.
I think it's possibly the result of the year. I think there's been a few that have come along during the year, but this looks good, I have to say. They're going about it the right way. It's a very plausible idea. So what they've done, this is a group at the University of Colorado, and basically what they've done is got a group of people with relapsing remitting MS, which is the commonest type, and that's the type that's characterised by recurrent autoimmune attacks on the myelin in the central nervous system with long periods in between attacks where the symptoms don't progress. And basically what they did was they ranked the volunteers in the study on things like walking ability, motor skills, cognitive scores, quality of life, using a very robust metric. And then they, having been nice to them and collected data about them, they then proceeded to wipe out their entire bone marrow and basically gave them high-dose immunosuppressive therapy, which is a polite way of saying that they would have been sick as dogs for weeks. But during the time, when they were being nice to them, they had collected a whole pile of stem cells and they reimplanted them with their own. So it was an autologous transplant. They reimplanted them with their own stem cells, having gotten rid of all the other stem cells. And their hypothesis was that that would then lead to no further, it was like a reboot, if you like, that whatever had gone wrong before would now not go wrong. And because it was autologous stem cells, they don't have to stay on the immunosuppression afterwards. And to get scores like they've been getting, where they actually get improvements in the disability scores and improvements in the quality of life scores in an overwhelming majority of the volunteers. I mean, this is just huge. They need to now prove the next thing, which is, it's one thing to say, okay, we've seen this association, but we don't know if it's causal. And so what they're doing is they're following these people over time to see whether or not the myelin is actually regrowing. And if it is, then that is at least relapsing remitting MS for certain patients who are fit enough for this incredibly strenuous treatment. But this is a massive step forward in this treatment, and this could be the year that we started to get a real grip on MS.
Mm. So is the immunosuppressive therapy, was that just to test the hypothesis, or is that actually a part of the treatment?
No, it's part of the treatment, and so what they're using is the same protocol that they use before they give non-autologous stem cells for treatment of various types of lymphoma and stuff. So there is well-established protocols they can use for basically making sure you have no rapidly dividing cells left in your bone marrow, and then they replace them with either your own in an autologous one or somebody else's, or some ones from sourced from another source.
Gotcha.
Yep.
It's a grueling treatment, though. I mean, what was it, 3 months, did you say, where they've got no immune system being rebuilt back?
Yeah, basically they have to be in isolation for at least a month once they've actually had the transplant, and then that takes about 6 to 8 weeks to wipe out the immune cells prior to that. So it is, as you say, it's pretty grueling, and it's sort of like, killing them to resurrect them in a new form, immunologically speaking.
And this was only done with, I think it was 24 volunteer patients.
That's
obviously,
right.
it's difficult to get a bigger sample size.
Right. And this is as I frequently have to explain to people, if you have a real treatment and it's not a complete waste of time doing a study in 24 people if you have a very plausible hypothesis and you have a big effect size. And this is about as big an effect size as you can get. It's about as convincing as you can get. 80 and 90% relapse prevention and improvement in their disability scores is incredibly robust. So this is a big effect size, and it's a very tightly done study. So the small numbers don't really weaken your faith in the plausibility of the results in that respect.
It's very cool. It's almost as cool, and I don't know why I like this story so much, but in North Carolina State University, they made a machine that has been puking vanilla pudding. They made a puking machine to study how norovirus spreads through human vomit. I don't know why I like this story so much, but it's so cool.
I think it's obvious why we like this story, because have you seen the picture on the story?
The uncanny valley picture.
I remember talking about this story and just looking at the machine, just going, "What the hell? They make that any more creepier than it already is." No,
there's no need to put a face on this machine,
But it looks like it was made from just clay that was just lying there for some reason.
It actually looks like a cast of my face last time I had norovirus. It's a sad and resigned look.
No, it actually looks dead. It's like
That's how you feel. That's how you feel. I can honestly say my oldest son, our first baby, had his first playgroup and brought home norovirus, and my poor mother-in-law came to the door of the house, knocked on the door. We opened the door, told her to go away and run as fast as she could because we're all as crook as dogs, and she still got it. And it makes a lot of sense, because when you look at their study, they were saying that you can aerosolize from a single vomit with their little machine, you can aerosolize 13,000 virus particles. Now, norovirus is just about the king of infectious viruses. You only need 20 active viruses to get someone sick.
Wow.
And one vomit aerosolizes 13,000 in viable form. So it's not surprising that, you can get sick from the slightest thing with norovirus. And it is just the worst type of gastro that you can get.
I think, if anything, it's more surprising that it's not more common. If it only takes 20 particles and a single spew is going to put out 13,000, I'd almost expect everyone to get it down. Particularly, I think it's known as the winter bug because it
you
thrives
don't
in
have
cold.
children, do you?
No, I don't.
Everyone does get it.
But I imagine because it's relying on the symptoms of the sickness to spread, you're already symptomatic by the time you're spreading it, at which time, because you feel crap, you're already at home, I guess, a lot of the time, unless you're, wandering around throwing up in the street.
Going to the chemist to buy...
Well, I know, I mean, obviously without getting too graphic, but I certainly know that when I've been in this sort of situation, I've not wanted to stray very far from the bathroom at all. Certainly wouldn't have gone in the car to the...
No, that's only the start of your problem, because when you get norovirus, you might get there in time, but you don't know which end to point at the toilet.
That's it. That's exactly right.
Well, I'm glad Lucas wasn't going to get too graphic. Thanks, Mick, for throwing that one in.
Rick can be... He has the professional sort of air of someone who is a professional.
We're just amateurs.
I've just been there one too many times.
Well, I mean, this virus is so nasty that the actual virus they used in the experiment wasn't actually norovirus. It was a different virus, a bacteriophage.
Which had similar, because they just needed to measure the effect.
Well, they needed to measure the infectious dose and all the rest of it. So, it was just a bacteriophage that's harmless, but they used that as a stand-in for norovirus because they didn't want to put
There's
norovirus.
certain ethical considerations with aerosolizing norovirus.
I think they're OH&S considerations for the poor people that actually built the machine.
Yeah.
But I just love that they even built it, and that it's a sort of test that you need to do. It's good science, and it's just a concerning looking machine.
Why they chose vanilla pudding as the stand-in for vomit?
What would you prefer?
There's no carrots in vanilla pudding.
What is it about vanilla pudding that's
disgusting? That's
I mean,
it'd be the right amount of viscosity and that sort of stuff that you'd want, I guess.
But there are different sort of
This is true.
consistencies and viscosity levels of anyway.
Maybe this is a stage one.
Well, it actually could have been. It could have been something that, I mean, the consistency of the pudding could have been one reason why it created a good aerosol.
Yeah.
Why not yogurt?
Maybe the yogurt would have killed the... Oh, no, it's virus.
Yeah.
Well, the probiotics would get rid of the unhealthy stuff.
I think we'll let them figure out the next stage of this test themselves. I don't think we need to offer any suggestions.
So they've not asked us.
Can I say, I don't want to see
Don't need the machine. They can just have my child.
And you actually mean that, don't you, Penny? It's like this.
At the moment, yes.
I'm visualizing Penny literally arms outstretched,
both of
holding
them.
her baby, going, take it from me.
It vomits. You can have
It's
it.
not vanilla pudding either.
No.
But again, the problem there, as Mick would say, is you don't know which end
end?
point. So at least this machine, they can control that.
Can I just say, though, I'm not looking forward to the logical extension of this research.
I can see where you're going with this already.
I don't need to say anything.
So yes, that's true. They imagine doing
the clay
the
model.
guy who makes the clay model of the other. Yeah.
and what vehicle do they use to
I think that's not as groundbreaking a machine. There's already plenty of Mr. Whippy machines out there that they could use for that.
When they're sort of low on the... Anyway.
Let's move on, shall we? Because, Shane, I know you want to talk about the bacteria that... there are 4 types of bacteria that seem key to stopping kids developing asthma, for want of a better way to put it.
Yeah, look, this was a story we did earlier in the year only a few months ago, actually, now that I think about it. It was a Canadian study, and they were looking at the faeces of 319 children. So, God, well done, guys. You clap on the back for that, because I wouldn't have done it. Anyway, and they were looking at, I think, basically the risk of asthma. The link between the bacterial community and the risk of developing asthma. And basically, if you had a mix of 4 certain kinds of bacteria, you are less likely to develop asthma as a child. And they've linked this back to the so-called hygiene hypothesis, where we're all just too damn clean. We're not, we're using hospital-grade disinfectants to clean our homes, and we're not exposing ourselves to nearly enough bacteria that we need to, and therefore we're basically growing up in sterile environments.
But also, like, the first 100 days were important for developing this microbiome, aren't they?
Yeah, that's right. And then after that, I think it basically normalizes. After that, it doesn't make much of a difference anyway.
But what are the implications then? Should we be giving doses of these bacteria to newborns or something? Or?
Well, this is the thing, isn't it? Is that this again? It's associated, but is it causal?
Yeah.
Well, we don't know, and I guess that's what the next.
So who's going to market the first FLVR supplement?
Surely it's already out there. Surely you can get it online now and have it. You just got to provide your credit card, and that's it. You're done. You're immune.
Yeah.
Well,
We've cured asthma. Done.
Cured it. Done.
Well, what I like is that they also did it the other way around. They actually used these results and developed an animal model and replicated the results, which I think it's usually done differently. So,
but as we've said, humans are not mice.
No, but the point is that they backed it up anyway. They
backed it up with an animal study.
Yeah.
But what I do like about this is it's once again showing that our microbiome is not just a matter of breaking down food and giving us energy. It plays a role in our immune system, in our moods and our thoughts as well. There's a lot more to be said for the microbiome than previously we've given it credit for, I think.
I remember we were doing a microbiome story every week.
Yeah.
It was, and as interesting as it was like, for God's sake, can we stop? No, but it's very interesting because it is actually a lot more important than we ever gave it credit for. I remember when I was doing my PhD, there were a few people working on this, and even back then it was pretty clear that, hey, this is actually a lot more important than we give it credit for. And,
yeah. And probably the last of our main stories, I guess, Shane, before we move on to some of the rubbish that was in the media. Shane, you're a big fan of this way of engineering viruses to fight bacterial infections.
Yeah, look, I don't know if this is a big story in terms of, it didn't really get a lot of headway because it's.
I think it's more a proof of concept, really, wasn't it?
It's also something that people don't really get.
Mm.
It's very hard to explain to someone who doesn't understand how viruses work, or and how they can be used against bacteria. Basically, so for example, if we use the human gut as an example, we have a lot of bacteria in the gut, but I guarantee that the viruses in the gut will outnumber the bacteria. And they're basically to infect bacterial cells. And what's really nice about them is that a lot of viruses or bacteriophages will only infect one kind of bacteria. They're that sort of specific. Well-honed. Yeah. And so scientists from MIT have used this as a proof of concept and said, well, why can't we engineer viruses to target certain kinds of bacteria? And we know the genetic sequences of a lot of bacteriophages very, very well, and we can manipulate them very easily. So they developed a new technique where they would manipulate these, what they call the gene scaffold, in another cell, which they also understand very well, like a yeast cell. They understand the biology of it very well. They know how to take things in and out of it. So they basically maintained these artificial chromosomes in yeast cells, and then they're able to splice in genes in and out of the virus genome as they saw fit. And what they were doing was targeting the bit that attaches to bacterial cell walls and injects its genetic material. And you can do that because virus genomes are very modular. Like you can basically, there's a bit for the head, there's a bit for the middle, and there's a bit for the tail. And the tail is the functional bit that sticks. And so you can change the genes. You can change those genes in and out and say, okay, now I want you to target this kind of bacterial cell. And they could do it. They could actually. I think originally they took a virus that only infects E. coli cells, and they changed it so that it could infect other species of bacteria. which I think is really, really cool. Like
That's great.
It hasn't been approved, obviously, by the FDA as a therapy, but I can see a point.
As we said, it's a proof of concept.
Yeah.
No, it's potentially extremely helpful, actually, because the thing with the microbiome is that it's vast and teeming, and it has some unworthy citizens. And if you could target these viruses to just go in and just sort of take out the baddies and literally leave everything else
like the
in place. Exactly. And even more because again, things like Crohn's disease, ulcerative colitis, they've often postulated that they might be due to pathogenic strains of particular things. So if they could just close that circle, if they could say, okay, these are the strains of E. coli. If you have someone with Crohn's disease, let's just go in and take out these baddies. Because, again, Crohn's disease is a relatively common type of inflammatory bowel disease. It causes a huge amount of, misery and hospital stays and gut surgery and everything. And, if you could have something like this, which could, very selectively and with very minimal side effects, just be able to go in and take out the offending organisms that were exacerbating the disease. It's a huge, huge advantage.
It's almost like the sort of thing that you'd be able to vaccinate yourself against certain infections. So if you're showing some signs of something, take a pill. It's a virus. If it doesn't find those bacteria, then not a problem.
It can't infect you anyway, yeah.
Yeah. So,
another one where this would be potentially really helpful is C. diff, where you've got this overgrowth, the Clostridium difficile, or a lot of people call it difficile. I'm an old Latin scholar, so I call it difficile, because you have to use a hard C if you're an old Latin scholar. But anyway, if you had people in ICU and you don't want to give them lots of antibiotics and so forth, you could just chuck a bit of these viruses down their nasogastric tube, and you could selectively take out the C. diff that was causing the trouble. And it's potentially, that's much nicer than a fecal transplant to sort of.
Yeah.
Yes. I think there's no argument there. All right, well, these days, it seems more than ever, perhaps, there are bad studies done or studies that are badly reported in the media, things that are just plain wrong. And this year was certainly no other. Let's have a look at the year in BS, shall we? So there was a study that showed that there's no such thing as a totally heterosexual woman. They thought that every woman is somewhat bisexual, and that turned out to be something of an oversimplified conclusion, confusing attraction, arousal, orientation, everything else. That was a load of crap. There were reports that... Well, speaking of crap, because there were reports that seemed to suggest men's beards were full of poo, and I remember you were very upset by this theory, Shane. Those stories were, as I said, full of crap. It started off with a news crew taking swabs of a few beards, sending them off to a microbiologist who found, among lots of other bacteria, a few types of bacteria that can be found among lots of other places in faeces, which was enough for headlines. It was enough for quick grabs on tabloid and television and media. And Penny, I'm sure you remember that right when you bought a new barbecue, the World Health Organization declared that meat causes cancer.
We're all going to die.
Turns out what they really meant was that
they're utterly certain that it causes a little bit more chance of cancer.
It might increase your chance by a teeny tiny insignificant amount.
But they're very certain that it will.
Well, they're reasonably certain that it will anyway.
Right,
I don't think it was even in the absolute.
I believe the article said they were totes certain.
Totes. Totes and mazeballs.
And these results are totes for real. Yep.
They're fully sick, bro. Can we stop this, please?
Yes.
No.
Please.
So the take home from that is don't worry too much. You can still use your brand new barbecue. And Mick, there was one study that I know annoyed you a lot, which tried to show a correlation between fluoridated drinking water and hypothyroidism.
What?
Yeah, it was a dog of a study. It was a classic example of bad science done with a conclusion in mind. And so
it's
this
a
was
fishing
the one
expedition.
Look, it was even worse than that. It was basically coming up with an idea, like back in the 50s, because fluorine competes with iodine in high dose, and it can block uptake of iodine by the thyroid gland. So back in the day, back in the 50s, they were using high dose fluoride as a way of trying to treat hyperthyroidism, in other words, lowering the thyroid function. So these guys have designed a giant long bow, and they've decided to look at the difference between regional variation in fluoride ingestion in the UK and the incidence of hypothyroidism or underactive thyroid. So they've drawn a very long bow, they've assumed causation, and then they've done the wrong sort of study to try and show it. Because what they've actually done is just a cross-sectional observational study with enough data that they could have controlled for a whole lot of things, but they chose not to. And then they basically tortured the data until it screamed. And then they were able to come up with the conclusion that they wanted, which was that adding fluoride to your water supply gives you hypothyroidism. And I don't know if anybody remembers what someone who grows up with hypothyroidism is called. The original medical term for someone who has a low IQ, a cretin. So the cretins in this case were the people who cooked up this awful study, but more so the reviewers who didn't kill it in the cradle.
Yeah, this got published in a relatively respectable journal.
It got published in a BMJ Journal of Epidemiology and Community Health. Oh. And a few of these flaws were not pointed out until the study was actually published and heavily reported on. And it was just a dog of a study. It was a really badly done study that should have been killed off in peer review, but somehow didn't. And it enabled the tinfoil hat brigade to start coming out again and saying, I
haven't heard much from the fluoride people recently because they've been taken over by the anti-vaxxers.
They're still there. They're still there, believe me.
Wow. They're one and the same thing, I think, in many cases.
Well, they probably are, yeah. But
I've written 117 articles now for the Conversation website, and the one that I wrote about fluoride activism is still the number one most commented upon by a large margin. Dear God.
And I'm sure they're not all supportive comments, are they?
Some of them very helpfully point me towards other websites that I can use to properly inform myself of
I bet they do.
what's going on. Our lizard overlords await.
Well, on that happy note, thank you, Dr. Mick Vag, for joining us today.
Never less than a pleasure, Ed.
Where's the best place for people to find you on the net? The Conversation you just mentioned then?
Yeah, the Conversation these days is probably the best place to find me, and I bob up in the columns there occasionally.
And, of course, thank you, Penny, Shane and Lucas.
No worries.
Thank you, Ed. Said.
And we'll have all the links to those stories on the web at scienceontop.com/211. Merry Christmas and happy holidays, everyone. We'll be taking a short break, but we'll be back again probably around mid-January. But in the meantime, we'll have our bloopers episode up, so keep checking scienceontop.com for that embarrassing schmozzle. And I'll also put up an interview that I did with British comedian and co-host of the Infinite Monkey Cage podcast, Robin Ince. So thank you everyone for listening, and like I said, we'll be back again next year, putting science on top of the agenda. Join us then.
Around a quarter of the school's 270 students have been struck down by the itchy infection. The school says children can be enrolled without being immunised. It asks parents to be tolerant when interacting with those who have a differing opinion, especially when it's an opposing understanding of child immunisation.